All right, so welcome to my lab practical walkthrough. It should be pretty quick, pretty easy breezy. These are really just the very most fundamental basic skills in a microbiology lab setting. I just really need to make sure that you guys have them all locked down before we move on later in the semester to more complex assays and more complex concepts. This is approximately what it's going to look like when you pull up. You got your broth and pipette area, garbage area, streaking area, sterile flame, it's mostly sterile flame, staining slide creation area, scope, paper. You will not have my wallet, unfortunately. So you guys aren't gonna have a whole rack of cultures. You'll just be handed a single culture tube and it won't have a label on it. It will be either E. coli or Staphylococcus epidermidis. So your first move is, even if the culture's already been mixed up by the person taking the practical before you, you probably give it a little tap and a few inversions to make sure those bacteria are uniformly distributed in this tube of broth culture. You guys should have already noticed this in class, but if you get a culture that is undisturbed, the bacteria will often just kind of settle at the bottom. My camera is not focusing. There we go. It just looks like that. So you want to make sure those guys are spread out throughout the culture. So now you got to grab yourself a P200 pipette. Remember, the name we use for the pipette is the largest number on top. It's labeled right here. So 20 to 200 is just a 200. We'll give you an amount to set it to. Remember that on a P200, the top number is the hundreds place, the tens place, and then the ones place. So if you wanted to set this to, say, 150 microliters, these are in units of microliters, you could just spin it using this black part of the pipette. Don't spin it using the top or else you'll risk messing with the calibration of the pipette. Got up to 150, there we go. The tips, I've had a lot of students actually struggle to match the tips with the correct pipette. Just know that the boxes of tips are labeled and the tops of the pipettes are labeled, so you can just find matching sets. I know many of you guys had to take your intro to biolabs remotely during the pandemic, so no worries. Just make sure we're square on this moving forward. So we should open up this box. We don't want to just leave it wide open too long. Lower risk of contamination if we leave it closed as much as we can. So to put the tip on the pipette, we just gotta kind of crunch it down on there, give it a little tap. It sticks on there. Notice that the tip does not come up over this silver part. Right, it stays down there just on the white end of the pipette, just fine and dandy. To release a pipette tip, also remember, don't use your hands. You should never touch the pipette tip with your hands. You can put it in the solid waste basket by releasing it using this button right here. Easy peasy. Please leave your pipette tips in the solid waste, not like a liquid waste beaker like this, because then when I'm cleaning out the lab, it takes me hella long to get these solid tips out of the often very purple liquid waste basket. It's not fun. Please don't put your tips in there. Put them in there. So to mitigate risk of cross-contamination and also just to, evaluate you guys on your basic lab skills, one thing we're gonna have to do is get everyone to make an aliquot of their broth culture into their own little tube. If you've never heard the word aliquot before, as a verb, it means to take a subset of a larger amount of liquid and move it, a small amount of liquid, into its own little tube. You can also use it as a noun to say this is an aliquot of this, right? So pretty straightforward. Uncap this guy. Try not to set down the lid. Pipette in. Draw up a little bit of liquid, whatever amount we prescribe to you guys. Got to set it correctly on the pipette beforehand. Recap the mother culture. Pop it back into place, and now you guys got your own tube. You can pop that guy open. It should be just a snap cap. Dispense into it, and then you got your own culture for streaking and microscopy. So, easy. Don't forget to label this guy as well. Got my initials, date, medium, nutrient agar, and then what we did with it. This is the practical demo. So you guys should all be pretty familiar with this at this point. You got your loop, you got your flame, And the inner cone right here is the hottest part of the flame. You can leave the loop in there for a second. You should see it can get red hot, and then maybe pass the rest of the loop through the flame a little bit. You don't need the rest of the loop to get red hot. As long as the tip gets red hot, you should be good. Now give it a few seconds to cool. If we were to put this onto the agar right now, it would, you know, burn the agar, which wouldn't be super ideal. Who let my facial hair get like this? God damn, I gotta clean that up. That is weird looking. It's not good. So for the actual streak on the practical, what I'm looking for is for you guys to, you know what I'm about to say, capture a broad range of microbial densities. And as I've commented on, I think everyone's lab report, the best way to do that is by packing a large number of back and forths in your streaks onto the plate as you possibly can. I think we can see in the first week of streaking, when you guys just did, you know, a squiggly line with a squiggly line through it, with a squiggly line through it, the density of colonies we grew was more or less uniform, meaning there wasn't a big drop off in the amount of colonies we were spreading around every time we added a new streak. So we want to make sure we're starting with a lot of colonies and we're ending with just a little bit, just a couple single guys that we know are monoclonal from one CFU, right? So methods wise, what you guys would recognize this as is either the quadrant streak method in your textbook or the T-streak method in your textbook. Doesn't matter which one you go with. Remember, the end game here is just to get as many back and forths on the plate as you possibly can. All right, so feel free to stick specifically to the methods described in the book, but this is how I have, you know, usually streaked plates. Can start by doing sort of some long guys over here, and we go right through the area we did before, right? And we're back and forth and back and forth and back and forth and back and forth, and I'm trying to stick around the edges here. That way I leave some space in the middle, right? So now I'm doing, you can kind of hold it in the light. I don't know if you guys can see where the streaks are, right? But you can get a sense of how much area you have left. So it looks like I have a lot of space up here for my third streak. What's up? Yeah. So, it's doing all right. I'm trying to document this on camera so I can just post it in the blackboard. Right, so it's my third streak. Right, I didn't cross back into my first streak. We just went over that line in the second one once or twice, right? We don't want to make the density uniform by just scribbling over the whole thing multiple times. And then the way I did this, you guys can see there's a little bit of untapped space in the middle. So I actually have room for a little fourth streak right now. So we can just do this guy and come in here, give it a little bit. And I like making this fourth streak a little bit wider so that those colonies are unlikely to overlap if they grow up large, right? If I leave them in the incubator too long, for example. That's like the official Riley left streak method, but feel free to stick to the quadrant method described in your book or the T-streak method described in your book. So just grab 20 microliters. That's what it looks like in the pipette. It's not very much. It's just a little bit at the end there. And we can go ahead and pipette some amount onto the slide itself. That's 20. So we've only got a little bit of broth on there, which is advantageous for this purpose because we're going to heat fix it, right? So I'm trying to give it a little bit of a spread out. You don't want to get too aggressive with it and lose all of the broth right off the edge. If we just give it a little bit of action to get it spread out, we get more surface area equals more drying power equals faster heat fix. So we got this going, and then we don't want to burn the bottom of the slide because we need light to be able to come up from the bottom through the sample to our eyes, right? If we burn the bottom, it'll char and we won't get light to pass through there. So we're just gonna kind of give it a little bit of, not in the inner cone, right, in the outer cone, just a little bit of pass through. And we're having the sample side up, right? We don't want to drop the sample off because gravity exists. We also don't want to directly burn the sample, which will, you know, kind of mangle those cells we're trying to look at. Just quick passes through here. It's gonna take a couple minutes to get this thing dried, and that's all right. Getting more spread out, faster drying. You know the drill. You could conceivably get this a lot more spread out. Actually, mine's pretty concentrated. But this is looking pretty dry to me. We don't have, I actually didn't get a perfectly non-charred slide, but it's good enough, I think. All right, I need two hands for this. So I got to prop up the camera in portrait mode, front facing, like an old person recording their grandchild's sporting event. But we got our slide right here. We're gonna hold it over the beaker. We do not want to spill methylene blue on the on ourselves or on our working surface. That would be bad. So remember, uncap this bad boy. There's no bacteria in here, so we can kind of just drop that cap, and we're going to liberally give this guy some methylene blue. There is no, you don't want to like just dump the whole tube, because you know other students have to use this, but don't be afraid to give it a large quantity of methylene blue. I've seen some successful attempts at air drying with methylene. I've also seen some successful attempts with washing. The hazard of letting it air dry is that there might just be too much dye left on your slide, and the hazard of washing might be that you might wash too aggressively, and if your heat fixture wasn't quite fixed enough, you might not end up seeing any cells, so they could get washed off in the process. So what we're going to do here, because it's faster to wash than it is to air dry, so we're going to wash, and you just got to make sure that your cells are actually heat fixed onto your slide before you wash, so you don't wash away your cells. Got it? So this is just water. Right? This is DI water. Well, I'm not certain that it's perfectly deionized, but it'll work for our purposes here. Just give it a little rinse. And I don't know if you guys can see this, but if you look very carefully, can we see that on camera? There's a little bit of stained area, like where our heat fixture is, there's a little bit of purple on there, right? That's good. That's good news. So once we got most of the water knocked off, you can also just give it a little bit of lens paper. I'm gonna set some down so we don't just leave this on the working surface, but we're gonna get just a little bit of lens paper. We're just gonna give this guy some gentle blots. Nothing too serious. Don't want to harm our little heat fixed bacteria boys. You know, obviously they're not gendered, it's just canonically, they're just dudes, you know. Cool, so let's see, are you dry, sir? Not quite, a little bit more. All right, I'm liking this outcome, I'm liking this result. So we got a, can we see it? Can we see that? Maybe I'll put it on something with a background. Yeah, so you can see there's like a very slightly purpley area here. All right, moving on to the scope. You can make sure that we're on the lowest objective on this scope. It's a red lens that we are switched to. It's the shortest by height. Pop this guy into place, trying not to touch the side that has the heat fixture and the stain on it. We can just use the edges of the slide so we don't interfere with it. The on switch, as always, right there, should give us the light. And then remember, the light control is right here. You get more light going this way, less light. I don't know if you can see it, but it gets brighter and dimmer, right? You can also use this dude to adjust the light as well. And then the focus knobs, of course, the big focus and the little focus right here, which adjusts the focus, of course, in a big way and in a little way. You got these dudes to move your slide. See how this moves it around? So you're gonna wanna navigate this generally to the area that has the stain on it. I'm thinking about right here is where I thought it was gonna look good. And then I can already tell you this is gonna be way too bright, so let's dial that down a little bit. All right, so this is one of the two possible cultures you could see on the practical. Basically, let's mess up the focus here so you guys can see it. You basically start from the lowest objective, use the coarse focus to find you know, where there's purple stuff, basically some interesting stuff. This is not a gram stain, so the color is not indicative of identity. So you're gonna wanna move around a little bit until you find an area that has a lot of stuff in it. We can maybe zoom in. I like this area right here. So put it in the middle, because when you're zooming in with higher objectives, you'll zoom into the middle of the field. Move up one level. You see it's a little out of focus, so we can use the coarse adjustment knob to put it into focus. We're looking good. I'm still liking this area. It's got a lot of stuff going on. We can see the stain is here, although at this zoom level we can't actually see individual bacteria. Peel appears that, oh, okay, there we go. This is the third objective. We can see I'm still liking this zone. So we're gonna move to the, between the third and fourth objective, we're gonna take a brief pause here. Just so we're all clear. So when you're at the, this is the stage where you wanna add oil. So let's say you're on the third stage with your slide on here. Once you're ready to move to the fourth, move it halfway, right, like this. Don't close it. This is when you can get your oil. Put a drop right where the light is shining through on your plate, or on your slide rather. And then that's when you can close off the fourth. And once you go to the fourth, there is no turning back. Going to the fourth now. This is the zoom level. We should actually be able to see bacteria. It looks like we are a little out of focus, so we can use the fine adjustment knob to get back into focus. And look at these guys. Okay, so this is, interesting. So let's pick maybe a more populated region to zoom into real quick. I'm not super pumped about this density over here. Let's maybe try moving around until we can find, I'm liking this over here. This is nice. We got a lot of stuff going on. All right, so let's go ahead and actually I'm gonna put this in the center right here. I'm gonna move to the second lens. We are mostly in focus. Give it a little turn, looking good. And then third lens, remember, no oil yet. Third lens, no oil. And we're a little out of focus now. We're in focus. We're looking all right so far. And then we can give it a third to fourth. Give it a little pause. Give it some oil. Right there on the dot where you can see the light coming through. I actually just tapped the slide with the oil thing, so hopefully that doesn't disrupt our colonies. Ideally, you don't want to touch it. You just want to drop the oil on there. All right, and much like the last time, we're just slightly out of focus. We're going to very delicately turn the fine adjustment knob, and look at those beautiful boys right there. Aren't they gorgeous? So this one should be fairly obvious as to what we're looking at. This is a nice and pure culture too. Don't see a lot of cross contaminants. There we go. Yeah, good.